Good afternoon to all. Good morning and good afternoon to all. Welcome to panelists, speaker, and other participants to 10th SCAR Open Science Conference 2022. The Antarctica in Changing World. Hosted by NCPOR, National Center for Polar Ocean Research, Ministry of Earth Science, Government of India, Goa. So our session comes under the life science, and the session theme is Southern Ocean plankton diversity, food web ecosystem, and biogeochemical cycles. So myself is Rajnikant Mishra from NCPOR, and then Kerrie Swadling is from University of Tasmania, and Anil Kumar is from NCPOR, is the group director from our Southern Ocean program. And then Sabu is from NCPOR. And finally, Christina Weldrick is from University of Tasmania. So then I can go for this, the other schedule of information to participants. So each presentation for the participant will be 15 minutes. 12 minutes is presentation, followed by 3 minutes question, answer, and discussion. And after this oral presentation, there will be a e-poster. But e-poster, if anybody interested, they can come and can present at last of this oral presentation. Okay. Then there will be a housekeeping notes also. I would like to inform, question can be asked using the question answer box or chat box function in Zoom. And other information like session will be recorded to be made available to registered participants in the past event section of the events platform. The organizers are planning to upload the recording to this SCAR YouTube channel. If anyone doesn't want their presentation to be made available on YouTube, they can inform to scar2022@l1meeting.com before 12th August. So, and then now I would like to request to Dr. Sabu, please take care of for this presentation timing and all, and please inform us so that we can finish first and go for the next presenter. So I would like to invite the first presenter is Camila Cataldo Mendes, and the title is A Study on the Energy Content and Demographic Composition of Antarctic krill Swans in East Antarctica. So may I request to Camila, please come and join for the presentation.
Should I just start? Is that how it works?
Yeah, please start it.
All right then. Well, hello everyone. My name is Camila Cataldo Mendes, and this presentation is about a study that I did last year on the demographic and energetic characteristic of Euphausis superba swamps. We all know that krill, Antarctic krill, is key in some areas of the Southern Ocean. It supports ecosystem services by playing an important role in carbon sequestration, supports one of the main fisheries in the Southern Ocean, and dominates the food web south of the polar front in the East Antarctic ecosystem. Krill provides a link between primary production and higher trophic levels. This is why gaining knowledge about the energy availability and energy flow from mid-trophic to top trophic levels is critical for ecosystem modeling and management. Krill as a population is able to do all these things for one main characteristic of its ecology, swarming. We know that krill can form big random aggregations, that they have a particular function and a particular characteristic for different types of aggregation. Krill swarms are very variable in all sorts of speaking. They go from 10 to 1,000 plus meters in length and 5 to 100 plus meters in height. Swarms can be huge, as we can see one here being detected with an echo sounder in the picture. An interesting topic is that is not really resolved is what triggers swarming behavior. We know that are non-random, so there must be a reason for it. There are very good ideas around this topic, but we actually don't know for sure. It could be due to changes in the water column, such as temperature, chlorophyll, oxygen concentration in the water, light intensity, perhaps a combination of all of these. It could also be a behavioral response to the environmental changes of the combination of this. But what could be the possible reason for swarming? From a krill point of view, why swarming is beneficial. And for this one, we are not sure either. Swarming has been suggested to be a response to predator pressure. Like we all probably have heard before, the bigger the aggregation, the least chances to be eaten you have. But if a whale is engulfing entire swarms, it doesn't really sound too beneficial for krill. There is also evidence that swarming aids krill to locate oxygen-rich waters. As aggregation tend to deplete the oxygen in the water column, they must be moving to reach oxygen-rich water. Another reason is to increase fitness by reducing the energy cost of swimming. Krill within a swarm can maintain really high swimming speeds, like around 20 centimeters per minute. Together with this, they could also save energy for other things such as reproduction. It seems like we do know a lot about random details about swarms, but they are only have been studied in some regions, and the information for all of them is not equivalent, meaning that it's very spotted around the Southern Ocean. We do know a bit more about the physical characteristics of krill swarms, the ones that can be targeted with acoustic tools. At least we have settled with a couple of characteristics that can describe swarms. We know that things such as size, length, height, shape, and packing density vary from swarm to swarm and from region to region. Packing density meaning how close the krill are to each other within a swarm. The study of swarms has been focusing on finding a way to predict swarms, where they're going to be. And for this, scientists have tried to classify swarms by their physical characteristics. There's not a single characteristic that can describe swarms or their behavior. And because of this, there's a lot of research available in this regard, but not much in terms of swarm biological characteristics such as demography, what stages are conforming a swarm, morphological characteristic, or energy content of the individuals within a swarm. Swarms can be composed by different krill stages and by individuals on different of different size and in different proportions. And we can also know that the physical characteristic of swarms varies in accordance with swarm composition. However, we are yet to establish patterns on this regard. All we know about this is that swarm composition are composed by individuals of similar size. There is, up until now, no evidence of what's the energy content of whole swarms. So for this reason, I endeavored last year on a mission to explore what's the composition, or what's it, if we can find patterns in the demographic composition of krill swarms, and if we could be able to estimate the total energy content in them. To try and determine swarm energy content and demographic compositions, we first went all the way back and assessed the energy allocation in key tissues and individual krill using microbiome calorimetry to have a look into what is going on within different swarms. We then determined the makeup of more than 2,000 swarms from a demographic and krill morphometric point of view. So what are the stages and the size that are composing a swarm? And we did this using cluster analysis. Then we bring this all together and try to estimate content of whole swarms to finally be able to study the relationship between krill swarms and whatever is possibly triggering of with what variables is related. For any energy allocation, we found that, as it can be expected, gravid females had a higher energy content within the ranges found for other areas of the Southern Ocean. For this study, we found that gravid females had around 21 kilojoules per gram of individual. We found that comparing mean energy by individual can hinder a little bit the results, as it does not consider size. For this, I then standardized all the energy content data and compare it using units of grams of dry mass. And this showed that very unexpectedly that juveniles will actually have a higher energy content than other stages of krill. The study of energy allocation in different tissues show that digestive blads and gonads have a higher energy content in all stages, which makes sense as there is more lipid stored in these tissues. We also found a pretty cool relationship between the energy contents of female krill in gonad and muscle. As the gonad energy increases, the muscle energy content decreases, and also the muscle size. This could be, I think, a potential trade-off. Perhaps females are jeopardizing muscle tissue in order to have a greater reproductive output. This will make sense about that. As we know, if you can see in the picture here, we have a really big krill gravid female and one spent female that are of the same length. But the one at the bottom has lost around 34% of the body mass after laying eggs. There are lots of reproductive trade-offs of this sort reported in other crustaceans. In krill, there's only one reported on male krill, that it could be that it reduces lipid storage to use all incoming energy into reproduction. So this needs a little bit more study, but it makes sense. The cluster analysis group or data in 4 different clusters that we define as swarm types. The first one, cluster 1, we decided to call it type A, and its population, it was composed 59% by subadults, both females and males. Cluster number 2, or swarms of type B for Huron, was composed mainly by 26%, but subadults, and both male and female, and 25% of adult females. Swarms of type C, the ones grouped in cluster 3, were composed by 62% of spent females. In cluster 4, the one that we call type D, was composed mostly by gravid females, around 60%. Then we use this data or output to create krill boxes in a spatial area. So we created polygons around the swarms that we found during the voyage in order to allocate mean energy values to swarms that will fall inside each polygon. The total swarm energy was definitely an adventure to calculate. We had to make a few assumptions. The main one being that swarms were cylindrical at their highest height. We related the mean energy value of wet mass with swarm volumetric density and biomass, which we're estimating using the acoustic data to give an energy value to every single swarm detected in accordance to the type or cluster that they belong to. In terms of size, we found that swarms of type B, the ones composed by subadults and adult females, were largest in terms of height and length. In terms of volume, swarms of type B had a greater volume, however, and this swarm type was dominated by subadults, both females and males. Most importantly, amongst all of our results is that I noticed that larger swarms were not the ones with the higher amount of energy in them. Small swarms, in all senses, volume and length, the ones dominated by gravid females were the ones with the highest energy content. Not only that, but at the time of the year when the study took place, mid-reproductive season in summer, swamps of type D, the ones with the purple boxes on the map, dominated the area of the study. Whoa.
Camilla, sorry for interfering. You have just 1 minute left.
Yeah. We then looked into other variables that could be related to this trend, things in the environment. There was no relationship with environmental data and some energy. But an interesting thing is if we had data from a visual survey of baleen whales in the voyage area, and this survey only incorporated whales that were observed feeding on the target swamps. And when mapping the whale visual survey on top of the heat map made with energy values showed how baleen whales were actually aggregating in the vicinities of high density swamps, the ones corresponding of type D composed of rabid females. We could suggest from this that there's a relationship between swamp composition and whale aggregation. However, this was not something that we really looked into, and I did not do further analysis on this, but it's a good starting point to study. There are a few things that we can take from this study. The first one is that female krill invest more energy into gonadal development at expense of muscle tissue. Swamp demographic composition drives swamp energy content. And this study was the first one to quantify krill energy for all developmental stages in the same region and time span. And we attempted to quantify swamp energy based on swamp demographic composition and direct energy measurements. I would like to acknowledge all the people that helped in this study. And thank you.
Thank you, Camilla. It's for nice presentation on demographic composition of Antarctica krill, and you observed that krill, even with different temperature and environmental conditions, at the tropical as well as subtropical as well as at the coastal polar water. So let us see that is there any question? Yeah, in question box there is no question. And
can I ask one question?
Yeah, please.
A nice presentation, Camilla. That's a very informative. And what I understand, this krill community, we know that recently the Antarctic bottom water formation is seriously affecting in the Southern Ocean region. And this is one of the oxygen supplier, and higher oxygen conditions, krills are existing. Whether this has any relationship with the krill community existence? Did you hear my question?
I think she's muted.
Okay, so otherwise we can discuss in afterwards.
Yeah.
So can we go? So I would like to invite the third speaker, Yongli Jiang, and the title is Seasonal Species Succession Controlled by Zooplankton Grazing on Phaeocyst Antarctica in the Iron Limited Ross Ice Shelf Polyneum. Yongli, you are there?
I could not find his name in this panelist, so I don't know whether he's joined. He's not joined.
Oh, we are not finding in list.
He's we are doubting whether he's joined or not. His name is not there in the panelist.
I don't think he has come, sir, yet.
Yes,
Can
so
we
we may
move to
go
the
for
next
next.
So in that case, we can go to the fourth, that is again the video. That's a Wednesday Halter.
I will present live actually.
Okay, thank
Yeah,
you.
we have a video
Yeah,
of my other talk, but
Okay,
yeah.
and then your title is the impact of the Ampart Timasto Judy on ecology and carbon export in the Southern Ocean.
Yeah, correct. Can you hear me all right? And can you see my screen?
Yeah. It's audible.
Beautiful. Awesome. All right. Yeah, thank you so much. Oh, sorry. Here we go. Thanks for having me today. I'm pretty excited about presenting today and hearing all the other talks. They're pretty exciting. I will talk today about the impact of the amphipod Themistogordichodi on ecology and carbon export in the Southern Ocean. And this is a study of myself. I'm currently based at NIWA in Wellington in New Zealand, and Ben Scolding, he's based at Siren, Australia. And the study really started on the Solace voyage in December 2020 to January 2021. We sampled a few sites south of Tasmania, Australia, in the Southern Ocean. And our aim on this voyage was to quantify carbon export by the biological and physical carbon pumps. This is our team, so lots of nice people, and we had a really good time, especially over Christmas. It was really cool. But my focus as a zooplankton ecologist was to quantify or kind of study the role of copepods and gelatinous zooplankton in the biological carbon pump during the time. However, when we sampled the last site here, just south of the polar front, we saw something really interesting on the echo sounder. You can see the feature here. We just have a black thing marker on it. And not only in one frequency, but the different frequency, we saw this pattern appearing. There's this scattering layer popping up it was always between 50 to 100 meters and always appeared during the day. So during the night, it kind of disappeared, moved a bit down and merged with the other scattering layers during the night. But during the day, we could really see it. And then because we had so good weather and had a bit of time, here we go. Sorry. Just going back, my first thought actually was because, we south of the polar front, there might be euphorsids, so just krill. Oh, just krill, right? And also we had this minke whale hanging around for days before, which is not really a, quantifiable observation, but it's an observation. So I thought, okay, we have euphorsids here. But then, as I said, we had good weather and a bit of time left over. We could actually target these scatter layers with the RMT, the rectangular midwater troll. And we found heaps of amphipods. As you can see here, it's basically a single culture of amphipods. And this was actually a swarm of Themistogordichodi, which produced these scattering layers. We could target it actually 2 times, one time at 50 meters and one time at 70 meters. And as I already said, this swarm was only observed during the day, which could be because, with the ship's acoustics, the ship is moving, the swarm is moving, maybe it was somewhere else, but, we could only see it during the day. So maybe starting, I looked a bit what we know about Themistogordichodi and, what's there, what kind of knowledge do we have? And it is actually quite understudied compared to krill, especially. There are some reviews about it, and it's really good to have this information. Here, for example, this is a really good from Hafermans et al. And it calls Themistogordichodi the krill of the north. And actually north here means the northern region of the Southern Ocean. There are other Themisto species, but for the Southern Ocean, Themistogordichodi is really the only one. It is a food item for many species. As you can see here, it has been found in different the stomachs of all these animals close to the Western Arctic Peninsula. And it itself predates upon the most abundant prey. So for example, here you can see it feasting on a salp and even on krill. Here we go. As you probably not know yet, because you haven't heard the other talk yet, is that during Solace, we had a salp bloom in the polar waters. So salps, the Salpa thomsoni, were actually dominating really the upper water column in terms of biomass. Here, for example, there's some optical observations we did, and they're really pretty much staying up. There's a bit of a migration, but they're pretty much staying in the upper water column where Themisto is as well. So our conclusion was that potentially, or most likely, Themisto is feeding on the salps. It has been found somewhere else to feed on salps, so potentially has to be, of course, confirmed with the gut content analysis, but we didn't do that in this time. But potentially, it feeds on the salp. If you want to know more about the gelatinous plankton during Solace, there's a talk by Katie Baker tomorrow at 4:30 p.m. Yeah, she will talk more about that. What else do we know about Themistogodi? Its distribution is really circumpolar, especially as it already said in the northern Southern Ocean. Here on the right, you see a bit of an, let's say, outdated picture from the continuous plankton recorder. And you can really see that it's distributed quite, widely, especially in the Indian sector. Of course, this is because we have the most observation of the continuous plankton recorder in the Indian sector. But Themisto swarms are really opportunistic, just being in the right place at the right time, like in our case, really. And most studies we found are from the shelf or close to the subantarctic islands. So I really found basically maybe 2 handful of studies that just focused on Themistogodi in total since basically start of publications. So it's really understudied. So here's where I found some records from. For example, there's some published stuff. The red ones are published. Then I found some data as well from the orange ones. I just added here. And then there are really few from this open ocean. So most of them are really close to the islands, for example, from the lower FX voyage and Broke voyage. And also in our case, in this study. And actually for our study region, this is the really first, to my knowledge, the first record of an amphipod swarm. So maybe the question is better, what don't we know about Themistogodi? There are so many knowledge gaps, there's basically gaps, not much knowledge. There's potentially many genetic lineages of Themistogodi, which is not the focus of this study really, but it potentially makes things more complicated. There are many physiological rates unknown. So just proof, like checking if my rates I measured are correct, it's really hard to find even comparable rates. And then of course related to that, the role in the carbon cycle is quite understudied because we just lack data. For krill, it has been recently recognized that they're really important, or can be really important for the biogeochemical cycles. But as I said, lack of data for amphipods, we can't bring this review up for amphipods. So what did I actually do at sea? Because it was an opportunistic observation. It was all, thrown together within, a few moments that I had usually at sea. It's usually how I work at sea. I calculated the total standing stock carbon by using the RMT numbers, the rectangular midwater troll numbers. And then I calculated the carbon flux by the swarm. I focused here on the passive flux. So for the fecal pellet production, I incubated Themisto for 24 hours and then counted and measured the fecal pellets. And then I took carbon values from literature to calculate the fecal flux. And then for the carcass flux, I just used some amphipods in the CHN analyzer, and I measured the carbon content, and then used mortality rates from literature as well to calculate the carcass flux. I also measured respiration at sea, but because we couldn't really find the amphipods to migrate, I excluded it for now at least. And also I didn't consider things like excretion or molting, just because we have no idea. The results. Here are some examples from other regions of the Southern Ocean. You have some open ocean samples or records, let's say that, and then some South Georgia and Kerguelen Island. And you can really see that our study here is a bit lower than on the Kerguelen Plateau. The numbers are a bit lower. But actually, in terms of open ocean, this is the highest ever recorded for an open ocean location. If you look a bit closer to the numbers and keep the unit here in mind, there's like 3 individuals per cubic meter. So I would say quite low. If you look at the RMT, the echo sounder again, this looks a bit closer and a bit denser aggregation actually than just 3 individuals per cubic meters. But that's what we have. I guess that we might have, even though it was a targeted troll, it might be still an undersampling by the RMT, but as I said, that's the numbers we have. So for the fecal pellet production, I calculated some flux, but keep in mind I couldn't avoid them feeding on their own fecal pellets, so it is kind of conservative estimate. And also I didn't feed them during the incubation. which I should have had, but I didn't know at the time how actually the amphibole figure pellets look like, so I just didn't feed them. So the values we get here, I think, an underestimation. Also, here's the carcass production. Here's some other values I calculated based on the literature mortality rate I got from literature. And overall, this is around 6 milligram carbon per square meter a day. So this is a bit on the lower end, but quite comparable to what has been estimated for krill swarms. And also, this is about 12% of the gravitational flux, which sounds quite low, but still, if you keep in mind that the numbers are quite conservative and probably an underestimate, the figure pellet production is quite an underestimation. We still are really in a conservative area here. So overall, it's probably at a high potential carbon export by the amphibole swarm, but we need way more data and rates to back that up. As an outlook, Themisto has often been proposed as a winner of climate change. Sorry, it's quite late. I can't talk anymore. This paper here was published 10 years ago already now, so we hopefully get an update at some point. That with an increase of 1 degree Celsius in water temperature, that Themisto would increase or extend their distributional range, at least close to the Western Arctic Peninsula. And then also, as Galian just mentioned, SALP has been seen as trophic dead end, but with Themisto in the system feeding on them, potentially feeding on them, this is not the case. It might not be a case and might be connected to the rest of the food web. Of course, this is all work in progress, so still a bit to do with this study. And the question is, do we have the right gear to sample Themisto? Probably it's not enough to just look at RMT numbers. So what I want to do is to integrate RMT numbers with acoustic data, what we already collected, what's kind of done by krill as well, to calculate the size of the swarm, so have like a 3D picture, and also understand where they go at night. So my main takeaway messages here are that we found a Themisto swarm, at least during the day, would potentially feed on salps. This has been the largest swarm ever recorded for the open ocean, but I think it's still an underestimate, based on the RMT numbers. So we might have to integrate our RMT values with acoustics. There's a high potential in carbon export by the swarm. Of course, we need more physiological rate to do that. And then in future, hopefully, we have more targeted surveys that just focus on Themisto, because we need to understand their current and future role in the ecosystem and biogeochemical cycles. And if you have any questions, ask them now, or please send me an email here. Thank you so much.
Shall I ask one question? Mishra?
Sure.
Swanja, this is Anil Kumar.
Please, yeah.
How are you, Swanja?
Good, thanks.
Yeah. My question is that in Australian coastal regions, especially one southward moving current is there, Lewin current, right?
Sorry again, can you repeat that?
Lewin current.
Mm-hmm, yeah.
Yeah, so you are telling that the circumbodal current has a major role with this community distribution and production and all.
Potentially.
Yeah.
Potentially.
So what about the Lewin current, any role it has? Because it is affecting the Australian climate also.
Right. Yeah, I mean, we have the same with the East Australian current as well. I mean, we have more warm water coming to the Southern Ocean. So because the Themisto has a really broad thermal range, if the water temperature would increase, they would increase their range as well. That's why I showed the picture. So it makes sense for our part, the Indian sector of the Southern Ocean as well, that they would increase. However, I would say, because it's really patchy, the records we have on Themisto. So I mean, my study was a bit opportunistic and just luck basically. I think we just need more data to back that up and say, oh, they will increase in future, because just right now, just based on a handful of records, it's a bit hard to say.
Okay, thank you.
Okay. Any other question? Any other question from anybody? I'm seeing in that question answer box, there is no question. Okay. So, can I ask, it is this is why you observe during daytime, what specialty?
During daytime, sorry?
Yeah. You observe daytime, right?
Yeah.
Hmm.
Yeah, it's
Why?
really interesting because I mean, usually you would expect zooplankton to be down during the day, right? So that's why we were so surprised to find something like this, you know, the swarm happening during the day. But then because of the salps being up during the day as well, mostly, you know, at least in the upper water column, I guess, you know, that's why they were there. But yeah, it's really interesting to see that this feature is so prominent during the day.
So then in night, we cannot find in a surface or subsurface region.
We can, it's just, yeah, during the night we didn't observe them. I guess either it was because the ship moved in a different direction because we were roughly at the site, but because, you know, we sampled with the RMT, we were moving around, quite around a bit. And there's probably like krill, they're quite patchy and also specially. So just, yeah, I think we didn't catch them during the night, like with the acoustics. And we had
Because-
only time to use target with the troll during the day, actually. So
Yeah, because you know that it's like most of zooplankton is in a nocturnal. I mean,
Right.
in a night time, the density is more.
Exactly.
Compared to the, yes.
Mhm.
So what?
Yeah,
That is why
exactly.
it has some peculiarities of the daytime you observe.
Mhm.
I
Yeah.
mean, the density is daytime observed much more than the nighttime, or
Yeah,
how is it?
usually, just at this site, because we had salps and they were, you know, the most dominant zooplankton there, really. I guess they were just following the food, and why should you migrate down if there's so much food, you know, if there's like a buffet in the,
Okay.
you know, during the day even, then why should you migrate down? I guess this is why it is. But yeah, I have to really look into, I want to also look a bit more on other days because I know we observed this feature on the other side too, the first southern side. And we had salps there as well. So I think I would just compare the different acoustic signals
Then,
and
yeah,
see.
I'm sorry. Then another question is that is very interesting that you find in this towards polar front the density is more. I think in Kerguelen Island and all, if you compare to the STF and to the polar front, the density is higher. How do you link to this? Is it like the link to chlorophyll, or is this like linked to biomass or something?
Could be. Not sure yet. I guess also in our case, it was just a targeted troll, right? So we saw this and then we tried to really catch it in the water column, whereas the other sides, maybe their abundance is higher there too, but because they just did a kind of standard RMT troll, they didn't kind of catch it. So maybe it's just different sampling. Yeah, maybe it's chlorophyll. It's something, you know, we have to really see. I mean, on the other hand, Timisto is a bit of a higher level predator, so it doesn't feed on like the algae. It would feed on zooplankton and salps and whatever is a bit higher in the food level, like in the food chain. Sorry.
Okay, thank you. Thank you very much.
worries.
And now
Thank you.
we can go to the fifth speaker. If no more question, then can we move to fifth speaker? That's the Talitha Nelson. And the title is Antarctic krill swarms recycle and export iron and carbon in the southern ocean.
Yeah,
Talitha.
great. Thank you. Sorry, can you guys see the screen?
Yeah. Okay.
Okay, great. I'll get started.
Yeah, you
Hi,
can start.
everyone. Yeah, my name's Talitha, and today I'll be presenting a research project that I worked on as part of my honours thesis here at the University of Tasmania. And I looked at Antarctic krill swarms and their role in the iron and carbon cycles. So just as a bit of background context, the Southern Ocean is one of the largest high nutrient, low chlorophyll regions in the world. So what this means is that the Southern Ocean has high concentration of macronutrients, such as nitrogen, as we can see in this top graph here, but very low chlorophyll concentrations relative to these high nutrients, as we can see in the bottom of this graph here. And this is due to iron. So phytoplankton growth is limited by iron, and iron may also co-limit bacterial production with dissolved organic carbon. So key sources of iron may include melting sea ice, glaciers, deep winter mixing, or sediment resuspension. However, this iron from these sources are often very short-lived as they're rapidly uptaken. So it has been suggested that following these large uptaking events, that pelagic recycling, such as through bacteria and zooplankton, may further sustain iron in the surface ocean. So Antarctic krill are a type of zooplankton. they have this they have this circumpolar distribution around the Antarctic continent, as we can see in this image here, with quite large concentrations in the Scotia Sea. And they have an extremely large biomass as well, with estimates of up to 380 million tonnes. And krill also have this really interesting behaviour called swarming. So what krill do is they will congregate in these really tight and densely packed swarms. So we'll have very high concentrations of krill in just one small area. So because of all these reasons, it has been suggested that there is a high potential for the recycling and export of nutrients of iron and carbon from krill. However, we still don't really understand this role that krill play. So this leads me to my research question. So what we know is that phytoplankton are grazed on by krill, and then krill produce these really iron and carbon rich fecal pellets. So our first research question was, how fast do these fecal pellets actually sink? And we also wanted to compare intact versus fragmented fecal pellets. So multiple processes, for example, even if the fecal pellet is released within a swarm, because these swarms can be so dense and tightly packed, they're likely to be fragmented. But also, as the fecal pellets sink, they can dissolve on contact with seawater. So what this means is that they can then release iron and carbon, and this is known as dissolution. Or we may have bacteria which can then come along, they may attach to the fecal pellets and then remineralize the fecal pellets and release iron and carbon. So this is known as remineralization. So our second research question was, how much is recycled? But we specifically wanted to know how much is recycled within the mixed layer depth. So this is because this is where the nutrients will actually be available for phytoplankton. And then we also wanted to know, our final research question was, how much is exported out of the mixed layer following these two processes? So what we did was we collected fecal pellets from krill at the Australian Antarctic Division Aquarium, and then we used seawater from the Solace voyage for all of our experiments. And then what we did was we upscaled our data to our krill density and estimates, which gave us our swarm data and our oceanography data from the Tempo voyage in East Antarctica. So here's a quick video I got of the krill that I was feeding. So essentially I'd feed them a predetermined diet and then I'd collect the fecal pellets directly from the krill. So as we can see here, this krill is producing a fecal pellet. So I would wait, and as soon as that fecal pellet was ingested, I would collect that, and that would be my intact fecal pellets. And for my fragmented fecal pellets, I would either wait until they naturally fragmented, or they were ones that I had accidentally fragmented myself. So for our experiments, we first had a sinking rate experiment, which allowed us to determine how long the fecal pellets would sink, and then essentially how long they would spend in the mix layer. So this was very simple. It just involved timing the descent of the fecal pellets in a 1 litre cylinder of seawater. Then I had a dissolution experiment, and this allowed us to determine how much iron and carbon is recycled within the mix layer, and then we were able to get our total iron concentrations from this experiment. So essentially we had our filtered seawater here, and then the fecal pellets were placed on a filter paper within this filter holder here. So essentially what happened was this seawater was pumped through and then it was flushed over the fecal pellets, and then we were able to collect samples from this, which allowed us to measure iron and carbon. And lastly, we had this incubation experiment to measure our carbon-specific remineralization rates. So we measured oxygen concentrations because oxygen is often used as a signal for bacterial remineralization. And this allowed us to, we aimed from this experiment to determine our carbon recycled through remineralization. And we also got our total organic carbon concentrations from this. So onto our results, our mix layer depth averaged 16 meters. And for our sinking rates, we found that our intact fecal pellets sunk significantly more faster than our fragmented. So our intact would spend roughly 1.6 hours in the mix layer, whilst our fragmented would on average sink a bit slower and spend 1.9 hours within the mix layer. So onto our iron dissolution results. So we've got our results graph here. And what we have is our time in minutes on the x-axis and our cumulative iron dissolution in percentage on the y-axis. And here I've got, this is roughly 1.9 hours. So this is where the both types of fecal pellets on average would have sunk out of the mix layer. So the most important thing to take away from this graph is that we've got roughly 0.04% of total iron for intact and 0.03% of total iron for fragmented is recycled within the mixed layer. And so essentially over 99% of total iron for both our fecal pellets is then exported. For our carbon dissolution experiment, we have the exact same graph here. And again, the most important thing to take away is we've got these very low percentages of carbon that's actually recycled within the mixed layer. And over 90, like similar to our iron, we've got over 99% of our total organic carbon for both our fecal pellets is then exported out of the mixed layer. So I've got my remineralization results here. So I forgot to mention, but we did run this experiment twice. And so I had a 12-hour experiment first to just measure these short-term remineralization rates. And then I also had a 60-hour experiment to measure our long-term remineralization rates. So we've got our time in hours on the bottom and then our oxygen concentration on the y-axis. So as we can kind of see, our treatments don't really seem to be that much different from our blanks for both our experiments. What we were expecting to see was that our treatments would decline in oxygen if bacteria were remineralizing the particles. But we didn't actually find a significant difference between our treatments and blank. So this suggests that we weren't able to obtain a bacterial remineralization signal. There's multiple reasons for this. I also forgot to mention that all of our experiments were run at 1 degree. So this experiment was also run at 1 degree, and it is known that bacterial activity is dampened at lower temperatures. And also we ran this experiment with filtered seawater. So we're kind of aiming to understand how the bacteria in the krill gut that's already attached to the fecal pellets would remineralize the particles, but this could be why we weren't picking up a signal that there just wasn't enough bacteria in our experiments to make enough of an oxygen decline signal. So for all of these results, we then extrapolated them out to our krill swarms. So we had 3 different size krill swarms. We had our minimum one, which was 2,251 krill per meter squared. And then our highest one, which was likely a super swarm, was 33,000 krill per meter squared. So this swarm's quite dense and tightly packed. So if we assume that all of the fecal pellets released in a swarm, they remain intact and they don't fragment, we can get these values for how much iron and carbon might be recycled. So although we've got very small percentages of iron and carbon that can be recycled, our values are within the micromolar and the millimolar concentrations because our krill swarms are just so big, and same with our fragmented over here. But we can see there's slight differences between the between the iron for our intact and fragmented, and a bit more of a difference for our carbon. And then again, just reiterating that we've got over 99.9% of our total iron and total carbon concentrations as being exported. But when we do extrapolate these out to our krill swarms, we've got quite large values again of our particulate material that is being exported. So just for the sake of time, today I'm only going to discuss the recycling of iron and the export of carbon of our results. So I've got this graph here on the y-axis. I've got the micromolars of iron per meter squared per day. And this is where our study sits. And this is where another study sits that specifically looked at krill swarms and the leaching of fecal pellets for iron recycling. So our study is quite a bit lower. There's multiple reasons why this could be. Firstly, this study here, they used fecal pellets collected from wild krill, and they noted that these krill had been feeding on lithogenic particles, and lithogenic particles can be really iron rich. So essentially these krill were feeding on these particles and then producing these really iron rich fecal pellets, which could explain the differences. Also, they used a different leaching method. So they actually acid leached their fecal pellets, which likely means that they were able to leach out more iron than what we could with just our seawater leaching experiment. Oops. So we can look at our study compared to broader Southern Ocean sources of iron. So as we can see, our study is within a similar range of the estimations for blue whales and upwellings, but quite a bit lower than glaciers and sea ice. However, it's important to remember that glaciers and sea ice are rapidly depleted following the melting early in the season. So the iron's rapidly depleted by biology such as phytoplankton. So krill alongside blue whales and upwelling may be providing these sources of iron following these depletion events. So onto our carbon export. There's been quite a few carbon export estimates of from krill swarms. So as we can see, this study is definitely the highest. So there's likely multiple reasons for this. So firstly, all of these other studies used fecal pellets that were collected from sediment traps or marine snow catchers at depth. So essentially, they used fecal pellets that were actually in the wild, and they had likely undergone more fragmentation than our fecal pellets did. And lastly, we weren't able to actually get a bacterial remineralization signal. And in the wild, it's likely that these fecal pellets are being remineralized, which can dampen the export potential. So just to summarize, Antarctic krill produces iron and carbon-rich fecal pellets. And as the fecal pellets sink through the mixed layer, less than 0.01% of both total iron and organic carbon is released. And then as the fecal pellets sink out of the mixed layer, over 99.9% can be exported of iron and carbon. So just to conclude, Antarctic krill swarms export more iron and carbon than they recycle. However, large swarms may be capable of recycling 0.07 to 1.6 micromoles of iron meters squared per day, which may further sustain primary production. And there's all my references. And yeah, thank you. Is there any questions?
Thank you, Dalita Nelson. Is more informative about this Antarctica krill swarms, recycle and export iron and carbon in the Southern Ocean. So I'd like to ask the panelists or the audience for questions, participants for questions. And
Can I ask one question?
yeah, please.
So your nice presentation about the fecal pellets and export of iron and carbon, and from the mixer layer region you're telling about. So you told that the mixer layer is 16 meters. So which area it is? Usually Southern Ocean mixer layer is more deep, but you are telling 16 meters. Why it is so?
Yeah, that's a good question. So this is from this data was from the Tempo voyage in East Antarctica, which was near the Purds Bay. I can't really answer why it is so shallow. I did specifically only pick swarms between, I think it was -60 and -60.5 degrees. So I also linked my mixed layer depth up to that as well. So it could be that, you know, I think I only had 6 data points, so there's just not enough variability in that area. But yeah, good question. Other than that, I think that's something I should probably look at.
Okay, thank you.
Thank you.
Yeah. Any more questions?
Oh,
Dalita,
one
yeah,
question
please.
is there in the chat box, question and Q&A.
Yeah, it is there. So the question from Karen Westwood, fantastic talk. And then I'm just wondering how mixed layer depth was calculated. It seems quite shallow, so maybe the criteria was very sensitive. It might be fun to look at different methods to see how the export changes. Do you please tell something?
Yeah, thanks, Karen. So I use I can't remember the name of the calculation off the top of my head. I can definitely go back and look at that for you. But yeah, I think that's a good idea to look at different methods and to see how the mixed layer depth changes and then how that will impact our recycling and export. But yeah, I guess in those graphs I think that I showed before, we could see that our iron was really released within the first 24 minutes of the experiment. So I guess even if our mixed layer depth is a bit deeper, I don't think it should change our results too much. But yeah, something I should look at. Thank you.
Okay, then one from my side. This is regarding the experimental observation with this krill when you tried with the diatom and flagellates. So you feed up the diatom with 75% and flagellates 25%. So what exactly is, why it is 75% from diatom and 25% from flagellates? Is it like differable or anything else, or can we change in reverse, like flagellates 75% and diatom 25%?
Sorry, are you asking, yeah, I guess why we chose the ratio of 75 to 25?
Yeah.
Yeah. Oh, sorry. Yeah, so, well, I guess we're trying to mimic what was observed on the Tempo voyage. So one of my supervisors was on there and she said they just saw diatoms absolutely everywhere. So we're like, okay, we'll feed them this predetermined diet of diatoms. But then we also were like, well, I guess the phytoplankton community might not just be diatoms. You know, we could have flagellates or other phytoplankton species in there. So we kind of thought that what might be representative of, I guess, a majority phytoplankton community of diatoms, plus whatever else might be out there, plus considering the phytoplankton feed that, the krill feed that we had access to, that was kind of what we thought would be our best option.
Okay, it's just like some differential feed for the krill, or diatom is much more than the flagellates or something.
What's that, sorry?
Okay. Okay.
Oh.
So
Sorry.
yeah, if no more question, then can we go to sixth speaker, that is Goping Joe, and the title is Biological Physical Processes Regulate Autumn Prey Availability of Spiny Icy Fish Chanodraco wilsoni in the Bransfield Strait, Antarctica.
Okay, thank you. Good afternoon, everyone. I'm Qingyuan Yang, a PhD candidate from Shanghai Ocean University. I'm very happy to have this opportunity for us to share some interesting research with each other. My presentation today focused on the use of fatty acid to explore the effects of physical processes on the feeding of spiny ice fish in the Bransfield Strait. In the past few years, global warming has become a major concern for oceanographers, whether in reports or papers. The Antarctic Peninsula, the northernmost tip of the Antarctic, has become one of the fastest warming regions in the world. This climate change has subsequently led to rapid changes in biological habitats and a large number of endemic species with an uncertain fate. As oceanographers, we are very anxious to understand the response of organisms to environment from a food web perspective. This requires us to build a complete food web structure, but in previous studies, krill and tall predators have become of most interest. Our mission is to fill in the gaps, especially the portal fish. We are most fond of spiny ice fish as they are reportedly often found in dense krill swarms. It is noted that there are regional differences in feeding of spiny ice fish, which is mainly influenced by the composition of organisms in the water column. While analyzing our samples, we found that the sampling stations were located at different physical ocean conditions. So we divided the samples into 4 groups and named them WB, TI, CB, and ES. In addition to differences in areas, we found differences in body length between some groups. We used the MANOVA to test whether there was a difference in fatty acid composition and whether this difference were due to body length, areas, or their combined effect. The results indicate that the differences in fatty acid composition do exist, and that such differences are limited to between sampling areas. Once we got this result, we became very curious about the physical environment in which the sampling stations were located. We found that WB is under the control of TBW, a relatively warm and freshwater mass. CB and ES are under the control of TWW, which is on the contrary a cold and high salinity water mass. And the TI is in a relatively complex environment at the intersection of TBW and TWW. After understanding the physical environment, we were more interested in what kind of feeding differences existed between spiny ice fish in different environments and how these differences are generated. We can learn from the figure that the fatty acid profile of the spiny ice fish in WB as well as in TI remains constant, while the profile is similar between CB and ES. Also, there is a large part of overlap between TI and CB. This was further illustrated by anosome where the R values between WB and TI and CB and ES were close to 0, indicating a greater similarity in diet between these two areas for spiny ice fish, while the other two areas were more different from each other. In the following, analyzing differences in the contribution of autotrophs were observed in the feed sources of spiny ice fish between all areas. The WB was dominated by TBW, where primary productivity and chlorophyll A concentration from the continental shelf waters in the Gelatin Strait is high, and the stabilization of meltwater promotes the formation of a shadow thermocline and is conducive to the accumulation of phytoplankton biomass on the sea surface. This resulted in a much higher proportion of intake of autotrophic organisms by spiny ice fish than in the CB and ES sample. At TI, autotrophs from the Berlin-Schlossen Sea and the Gelatin Strait are present, and anticlonic eddies occur around Tower Island. Such mesoscale eddies may lead to deepening or shoaling of the upper mixed layer, which influence the vertical mixing rate of phytoplankton. This complex hydrographic structure resulted in a difference in the autotroph feeding proportion of spiny ice fish in the TI sample compared to the WB and CBAS samples. In addition, for these three areas, spiny ice fish samples showed different levels of feeding on plankton. Under the 2015 to 2016 El Nino climate pattern, krill biomass decreased in pre-communities in TWW controlled areas compared with those in TBW controlled areas, and the spiny ice fish selectively preyed on other organisms, reflecting highly omnivorous characteristics. The plankton larvae were carried near TI and can be trapped in the eddies. Individuals in eddies will have sufficient residence time to develop in this nutrient-rich environment. We speculated that at TI, considering its location at the confluence of the TBW and TWW, a complex source of zooplankton derived from the eddies, such that for spiny ice fish, there is not only an abundance of krill, but also a high biomass of other zooplankton to feed on. and the propensity for free or debris and planktonic or benthic sources differed between all areas. In our study, a strong benthic character was observed at CB and AS. This positive SAM phenomenon led to enhanced vertical mixing and enhanced nutrient transport from the seafloor to the sea surface. At TI, the Bransfield front and Peninsula front resulted in an upwelling region in the subducting region, which increased the benthic nutrient uptake by spiny ice fish. To make a long story short, we have accessed the diet of spiny ice fish in different water masses in Bransfield Strait using fatty acid biomarkers and describe the possible cases of these differences, reflecting the importance of hydrography in governing trophic availability of animals. Our study supports the idea that in Bransfield Strait, the feeding characteristics of spiny ice fish can adapt to the prey communities in different water columns, which may contribute to their success in climate change-induced habitat changes. Finally, I would like to share a part of what we are working on using fatty acid and stable isotope to explore the relationship between the feeding of spiny ice fish and environmental factors. We have found some interesting phenomena so far, such as the effect of temperature. Sorry.
Thank you, Indwen Yang. It's
Oh,
very interesting, your presentation on this biophysical processes and the ability of spiny ICPs. So can I ask the audience and the panelists to have any question?
Shall I ask one question?
Yeah, please.
Yeah, thank you for your nice presentation, Yang.
Yeah.
And you are telling about some eddy formation, the TBW and T1, that region. What kind of eddies you have seen there? Is it anticyclonic or cyclonic? How it is affecting this?
Sorry, I didn't
Eddies,
get your point.
Eddy formation in TW and T1 you are telling about.
The information of TBW and TWW.
Yeah.
The information, sorry.
Eddy formation, eddy. EDDY.
So sorry, maybe we can talk about this question, use the email.
Okay.
Thank you.
Okay, we can talk. We can discuss later on this regard. On this next, we can move to the seventh speaker, the same Whopping Joe, and this title is Summer Eugrigarine Infection of Antarctic Krill Euphasia superba relates to station, body weight, and diet of host in the Dumont de Urbilas Sea, East Antarctica.
Yeah, here.
Okay, you will be present?
Yeah. Okay, thank you everyone. And yeah, I'm Xiongyi, a postgraduate from Shanghai Ocean University. Today, the topic of my paper is investigating the factors influencing summer eugrigarine infection of Antarctic krill, Euphasia superba in the Dumont de Sea, East Antarctic. And the outline of my talk as follows. And Antarctic krill, especially Fascia superba, comprises a significant proportion of the biomass in the Southern Ocean and is an important food source for numerous species of whales, fish, seals, and birds. Therefore, in recent years, more and more studies have been conducted on Antarctic krill, but little is known about the parasites in krill. The right image shows one of krill endoparasites, gragrine. The most common gragrine in Euphausia superba is subterranean Pacific, which distributed around Antarctic with extremely high prevalence and intensity. In view of this, it's very important to carry out research on gragrines in krill. And gragrine enter the body of krill by trophic transmit. Then it will reproduce and grow in the digestive tract. Generally, gragrine are most abundant in the gut, followed by Hampton pancreas and finally in the stomach. Previous studies have shown that intensity of gragrines is affected by many factors, such as krill bodyiness, weight, maturity, density, and so on. On the basis of previous studies, this talk ends two factors, the gut wise and the stomach contents, and expose the correlation between them and the gragrine intensity. And Antarctic krill were collected in February 2019 in the laboratory. The individual krill were chosen randomly from each sample. Each krill was first measured for basic biological data, and the stomach and gut were then removed, respectively put on a glass slide and turned to pieces with forceps to observe under a night microscope. The data were analyzed using the generalized linear model. The result is shown in the figure. It can be seen that the intensity of gragrine is significantly correlated with sampling station, maturity, and body weight, but has no relationship with the gut wise. In the stomach content, intensity was only significantly correlated with dinoflagellates, but had no correlation with diatoms, cetacoflagellates, and copepods represented by heterotroph. Below, I mainly discuss the results for the stomach contents section. And first, whether the intensity of gragrine and copepods represented by heterotroph are related in Euphausia superba. In fact, in 2001, Trophic did an association of gragrine infection with dominant phytoplankton diet, as no gragrines were found in euphausias with copepods in stomach. Obviously, my result is different with his. And Kerrie et al. Used a DNA-based approach to measuring in situ feeding by northern krill and discovered synthesis of diverse species assemblage of gragrine parasites. Species of this gragrine general infect the gut lumens of multiple species of copepods and amphipods. Therefore, theoretically, it can be assumed that krill is on higher risk for being infected by gragrine with more copepods in the stomach. However, such assumption ignored the fact that gragrines that enter krill directly and those that reach krill through the copepods are differed in life stages. This figure shows the complete life cycle of the gragrine. Hosts are usually infected by ingesting infectious oocysts, which has a capsule. When oocysts ingested by krill, they immediately use krill's acidic digestive juice to dissolve the capsule and release free-swimming sporozoites, which then immediately attach to epithelium and turn to cephalin. Assuming the majority of eugragrine that enter krill while copepods are cephalin and or garment without capsule. When the eugragrine arrive in the krill's stomach with copepods, they should be digested and die with high possibility in the absence of a capsule. Thus, eukaryotes that enter krill via copyboats can be considered as feeding objects rather than parasites. Moreover, krill prefers to feed on diatom rather than copyboats during summer. So therefore, our data cannot support eukaryote infection is correlated with copyboat abundance in krill stomach, which should be further research in future. The second point is that the growing intensity is negatively correlated with dinoflagellates. And this is a significant discovery of the factors influencing the abundance of growings, which has never been mentioned in previous studies. Here we have two perspectives to look at the dinoflagellates in krill stomach. The first one is as a feeding item. During the observation under the stereo microscope, we found all krill showed an extreme abundance of diatoms and syncoflagellates or heterotrophs in their stomach, while the abundance of intact diatoms, syncoflagellate or heterotrophic remains were observed in the intestine, suggesting a better digestive utilization of krill on those diets. In contrast, abundance of dinoflagellates in the stomach is similar to gut, and even more dinoflagellates in the gut were observed than in the stomach for two krill specimens in station 24, suggesting a low nutritional contribution to krill and or a high digestive resistance of dinoflagellates to krill. So how do we extend this phenomenon and the negative relationship between dinoflagellates and the intensity of growing? Maybe we need to look at dinoflagellates from another angle, that is parasite. If dinoflagellates acts as a parasite, it may form a nutrition competition relationship with growing or predator-prey, thus hindering the reproduction and growth of growings, resulting that there was a negative correlation between the growings intensity and dinoflagellates. In fact, of approximately 2,000 known dinoflagellate species, about 140 are classified as parasites of crustaceans, and Jamie also summarized parasitic dinoflagellates have only been found in krill eggs in the book, the Global Diversity and the Ecological Function of Parasites of Euphasias. And these dinoflagellates can cause whitening of krill eggs. So there is sufficient evidence to prove that dinoflagellates are likely to be indoor parasites of entitled krill. In conclusion, the present study found that growing intensity was significantly correlated with krill body weight, maturity, and dinoflagellate in the stomach, but it was not significantly correlated with the gut-wise diatoms and copyboats represented by heterotroph. In addition, the abundance of growings varied across stations, suggesting that growing infection is as regional as krill feeding. And in future research, we still need to carry out more in-depth research on the growing in krill. At the same time, more attention should be paid to correlation between trophic infected indoor parasite and krill feeding. And finally, this paper also led us to think about whether dinoflagellates are feeding objects or parasites, which should be further confirmed in future research. Finally, thank you very much for this valuable opportunity to attend this conference. The study was supported by some funds, and also give my thanks to Dr. Sukal Kuchi for sending krill samples to us. That's all. Thank you.
Thank you, Sian, for a nice presentation on this infection of Antarctica krill Euphasias superba related to station body length and weight, and especially you say that it is like dinoflagellate is a parasite or a food. So what's your idea? What's do you think from your result?
Sorry,
Whether dinoflagellate is a food or parasite of the krill?
Dinoflagellate.
Yeah, you said in your conclusion that dinoflagellate, whether the question is food or parasite. What do you feel? What do you expect?
There is definitely say dinoflagellates as feeding objects or parasites. I just say it will play the functions that we not know, which need further research maybe in the future.
Okay.
Yeah.
Thank you. Any more question from a panelist or this audience? Kerrie, you have any comments?
Yes, I've got a question. Thank you. That's quite interesting work. Do you find, or do you think there's going to be differences depending on the time of the year and the life stage of the animals that you're looking at?
Thanks for your question, but I'm not very clear. The question mean. Sorry.
I'm just wondering if
yeah,
you did the work at different times of the year.
Oh no.
Seasons in Antarctica, if that would show differences.
It's just in summer. In fact, the research about Gregory is more concentrated on summer or spring. Very little research on winter or autumn. Yeah.
Yeah, that's what I would expect. And what about different developmental stages of the animals in their life cycle? Do you know if there's any work that's looked at across the different stages at different ages?
In fact, this question is, I think we need to do in the future, because now is we don't know that. Yeah.
Yeah. Do you have any thoughts about what it might be like? Could you make a prediction or guess?
Okay. From another krill species, the different season may it indeed have a difference between different seasons. But for Antarctic krill, especially we don't know the Gregories have any difference. That's maybe we need to do in the future. Yeah.
That'd be interesting. Thank you.
Thank you. Thank you. So now is all oral is over. So now there is a 4 numbers of e-posters. So can I ask the IT team, they can share these posters?
Mishra, there is a message from Spenja that Gerlian Verhagen is not giving the order.
Okay. But then other threes are there.
Only 2 more, no? That is by.
And Angela is not joined that, I think it seems. She's not joined. She's not in the panelist list.
Okay. So that means other two will be there, no? So
Gurlane is not giving, it seems. The
Gurlane
message
is
has
not.
been given.
uh-huh.
Then Guping shoe.
Think so, two posters. are
Guping
there.
Joe have a two.
Two, yeah.
So can those things can be displayed by IT team?
No, sir, we don't have the facility to display the e-posters. We only have the option of playing recorded talks for oral presentation.
Okay.
Then Guping shoe, she can share it, you ask her.
I think so. Is not joined. I think his or
I think
students are there.
that's
Students are there.
Okay, yeah, okay.
But,
okay. If they cannot join, then can we go for close this session?
I think so, because they can, the posters, they can see in that posters, e-poster space also. No? So accordingly, they can make comments also, if anybody wants to make comments.
Okay.
If all panelists are okay, then we can go for.
Okay, Kerrie, so what to do? We will close the session if.
Yes, I think we'll have to. I was told that we would be able to get e-posters presented, but I must have had the wrong information. I apologize.
I think if any other questions, I think we can take because If
any further questions or
questions
anything,
we can take.
But I'm not seeing anything in a question and answer.
Q&A, no questions, but from panelists or from participants.
Yeah. Okay. Any questions? I think it's okay. If not, then we can close this session.
Okay.
Okay. So then tomorrow again we'll have a continuation of two slots. So I'd like to ask Kerrie, would you please want to start tomorrow's session?
And at night, 7 o'clock tomorrow evening, our time. Forgot what time that is UTC.
Yeah, evening is okay, but the tomorrow session starts. I think your time is 19 to 21 hours. 19 to 21 hours. I think it will be late for you.
No, it's 7 p.m. our time, and it's 9
to 9. Yeah, 7 to 9.
Yeah, 9 to 11 tomorrow UTC. And our speakers tomorrow will be from New Zealand, Italy, and Spain, so they should all be able to attend. Okay,
then it's fine. So tomorrow then we'll meet again and two slots. One is from 19 hours to 21 hours of yours. I mean Australian time, and then other is 1 p.m. to 3. 1 p.m. to 3 midnight. I think. So it's not possible for you. So can that case I can, or from our side I can ask somebody to start this session, right?
I'll be here.
Okay. So for today we can close. Dr. Sabu is fine.
Yes. Yeah.
Okay. And so Anil sir, it's fine. We can close right now.
Yeah, I think everything we have discussed.
Okay.
You can close it. You want to take any photo session or something?
But no posters and all.
No, photo. I'm asking.
Yeah. Please. So can I request to all to just switch on the video? So Dr. Sabu will take one snapshot for this session. Switch on everybody their videos. Yes, still many are there. There's to be switch on the video.
That is yes.
Yeah.
So yeah, two more is there.
Yeah, other two guys are there.
Okay. Yeah, I'll take it.
Okay, then you can take.
Yeah, done.
Is done?
Thank you.
Yes.
Oh, thank you. Thank you to
Okay.
all.
See you later. Next
Yes.
session. Bye.
Bye. So we'll start again by tomorrow.
Thank you so much. Thank you.